Hi, this is Joey Heath, and in this video I'm going to be talking about the molecular mechanisms of growth in Star Wars. So the idea behind talking about the molecular mechanism of growth kind of came to my mind from The Mandalorian, the new Star Wars show that you can see on Disney+, particularly with respect to this adorable little guy. Before I move forward, I want to say that I'm going to touch on some spoilers for The Mandalorian. Nothing too heavy, nothing too plot intensive, but there are going to be a few things that if you haven't watched the show, you will see exposed in this lecture. So if you haven't had a chance to watch it, I highly recommend it. It's really good. So moving forward, I'm going to start by saying this is Jedi Master Yoda. You've seen him in several of the previous movies, and you know him. He's a wonderful Jedi, really powerful. And what you don't really know about Yoda is his species. So it was really surprising to, during The Mandalorian, see this little guy revealed. And I do want to say, though, while he's sometimes referred to as Baby Yoda, this is not Yoda. So he's technically, in canon, referred to as the child. So while he's called Baby Yoda, and it's okay to call him Baby Yoda until he gets an official name, he is not actually Yoda. So to kind of further hammer this home, the Return of the Jedi is when Yoda died, and The Mandalorian, where the child appears, takes place about 5 years after that. So pretty easy to say after seeing that the child is not Yoda. He's not affiliated with Yoda in any way. The only thing they have in common is that they belong to the same species. So there he is again. The basis of this lecture is going to be on the fact that the child is confirmed in the show to be 50 years old. And IG-11, an assassin droid, states that species age differently, which we know from studying developmental biology is a very true statement. So the basis of this talk is going to be the question, why is the child 50 years old and still a baby? So to talk about that, we're going to talk about beginning with cell growth and proliferation, intrinsic growth program, key regulatory pathways, and overall growth and timing. So starting out with cell growth. Cell growth itself is going to be considered an increase in the mass or overall size of a tissue in an organism. And while there are three different types, the two primary ones are cell proliferation or mitosis, the division of cells to produce new daughter cells, or cell enlargement, where the single cell starts to grow larger and larger. The most important one for the growth aspect that we're going to talk about with respect to the child is going to be cell proliferation. An important thing to consider when you talk about cell proliferation or cell division is how that process is going to be regulated to allow the normal growth, development, and functionality of the organs within an organism. So specifically talking about the cell cycle is going to be composed of G1, S, G2, and M phases. G1 and G2 have to do with normal cell growth. That's essentially when the cell is going to be doing what the cell is supposed to do based on the type of cell that it is. And the S phase is going to be where the DNA inside those cells is going to be duplicated so that each of the daughter cells, the two new cells that are produced from mitosis, are going to have identical copies of the DNA and essentially be the same cell, but just multiplied. So this process, though, the cell cycle, is regulated through the synthesis of these proteins called cyclins. So there are three different types of cyclins as indicated by this figure. We've got G1S cyclins, S cyclins, and M cyclins. These are going to be proteins that are going to regulate the progression through the cell cycle. And the expression of these cyclins is going to be dependent on whether or not the cell has enough material to proceed to the next phase. So as cyclin protein levels increases, cyclin is going to be able to bind to a corresponding CDK or cyclin-dependent kinase, which is going to form an active enzyme. So you can see in this figure here, this is a cyclin. It's going to bind to a cyclin-dependent kinase, and it's going to form an active enzyme that's going to be able to drive the cell cycle forward. And so as you can see in this figure represented by these red lines, the cell cycle is going to have different checkpoints that are going to ensure that the next step of the cell cycle will be able to happen. So once there are enough cyclin molecules present, it's going to be able to bind to CDK. And you can see that as the cyclin levels raise in this graph here, the cyclin-dependent kinase is going to be active. And then after all of the necessary steps have been made, the cell has gone through the cell cycle, after mitosis occurs, you see that cyclin-dependent kinase is going to be inactive as cyclin production drops off, cyclin's actually going to be degraded by different proteins so that the cell cycle can essentially be reset and begin at the next step. So now that we've talked about cell growth and proliferation, which is regulation at the single cellular level itself, we're going to talk about the intrinsic growth program, which isn't directly going to address why Baby Yoda is just a child or a baby at age 50, but it's going to talk about why him and any one of Yoda's species is so small. So the intrinsic growth program is something that's specified at early stages in development, and it's going to determine basic patterning of an organism and how large an organism can grow. And it's going to determine how an organism is going to be able to respond to growth stimulants. So essentially, this is going to tell you what cells within an embryo, and you can see this in the zebrafish fate map here, are going to be able to become a particular type of cell or a particular structure within the mature organism. So if you look at this fate map, and a fate map is essentially just a diagram of what the intrinsic growth program is saying, you can see that this little blotch of cells here in this earlier stage embryo is dyed with a fluorescent stain. And after that organism, that zebrafish, is allowed to develop for a little bit longer, you can see that those cells that were marked with that fluorescence in this little patch in the early embryo were actually developing into a particular structure here. So what that tells us is that we're actually able to determine, based on one particular cell, what it's going to grow into, based on the intrinsic growth program. And it's also going to lead into how large an organism can grow. So this is essentially a genetic pre-programming of what an organism can be in terms of its overall size, and what cells are going to become what and what size. And that's essentially saying that this little guy here is going to have an intrinsic growth program. So he's not going to become a 6-foot tall being. He's going to be about this tiny little size that he is, and Yoda is going to be just a little bit bigger than him. And that's going to say that these organisms are, in their DNA, are just not made to grow to be larger. So with that in mind, we're going to now move on to talk about a key regulatory pathway within the cell growth process, and how that could potentially lead into the actual act of why this little guy is 50 and still a child. So before we talk about the regulatory growth pathway itself, I want to mention apoptosis really quick. If you're not familiar, apoptosis is the process of programmed cell death, and that's going to be when a cell is damaged, stressed, or triggered by some sort of signal to say it doesn't need to be alive anymore, and the body can do okay without it. So it'll begin to essentially kill itself, break itself down, and no longer be a part of the organism. And the reason I bring this up is that it's really critical, as it is one of the eventual outcomes of the pathway that we're going to talk about in the next slide. The regulatory growth pathway that I keep bringing up is called the Hippo Pathway. And while it doesn't have anything to do with a hippopotamus, it does have to do with a protein called Hippo that we're going to touch on in just a second. So as you can see here, the Hippo Pathway was first observed in Drosophila, or fruit flies, and it also has been seen in vertebrates and other mammalian cells. The Hippo pathway itself involves a cascade of serine threonine protein kinases, of which Hippo is one. So when you look at this, this is going to tell you what serine threonine are. Those are just amino acids that compose the enzyme. But a kinase is going to be an enzyme that transfers a phosphate. So in this case, the kinase is going to facilitate the transfer of a phosphate from ATP to a substrate. And you can see that substrate gets phosphorylated there. And the term cascade means it's going to be moving down and down and down. Things are going to continue to be phosphorylated until the change in gene expression is actually affected. So the Hippo pathway itself, its activity is going to regulate the inactivation of a transcriptional coactivator called Yorkie. And the Hippo pathway in general is going to be responsible for either limiting or activating the size or number of cells that are in an organ. So the Drosophila Hippo pathway in its inactive state is going to occur. The inactivation of the Hippo pathway is going to occur because of mechanical stress that's perceived by the cytoskeleton or different inputs from other growth pathways. And that's the inactivity is going to allow this transcriptional coactivator Yorkie to translocate into the nucleus. And once it gets in there, it binds to a transcriptional coactivator that you can see here represented in blue. And that complex is going to bind to the DNA and induce the expression of two main types of genes. So the gene types that are expressed as a result of the Yorkie translocation is going to be the cyclin genes. And this is important because we just talked about what cyclins do. They're going to drive the cell cycle forward. So by inducing the expression of cyclins, it's going to favor proliferation. And additionally, specific microRNA genes are going to be made. And if you're not familiar, microRNA is going to be a small transcript, very, very small piece of RNA that's produced that's going to bind to another transcript and essentially cause it to be destroyed. So the specific microRNA that's produced by the inactive Hippo pathway is going to bind to genes or transcripts of genes that favor apoptosis. So essentially, by inducing the expression of these specific microRNA genes, they're going to halt apoptosis or prevent it from happening. So the overall outcome of the inactive pathway in Drosophila is going to be cell proliferation is going to be promoted and apoptosis itself is going to be suppressed. This is going to lead to cell division. Now, when we talk about the active pathway, it's going to be activated by growth inhibitory signals. So we want those cells to stop dividing. That's going to be growth inhibitory signals. It's also going to be inactivated by cell-to-cell contact. So when cells start to come really, really close to one another, the organ knows, okay, it's time to stop dividing. It's time to stop pushing the developmental process forward. And they also could be signals from polarized cells. So in this case, epithelial cells will get either a basal or apical side, and one of those two sides are able to release different particular signals. So once cells start to get polarized, they need to start dividing differently or not dividing, and that's going to start to activate the Hippo pathway. So this cascade, this is the cascade, the kinase cascade that I mentioned earlier, and you can see these different kinases. Here's Hippo, it's one of interest, but what you can see is that this protein, warts, right there, WTS, is going to have a phosphate on it. It's going to be phosphorylated through this cascade. Warts is going to phosphorylate Yorkie, and that's going to cause it to be degraded in the cytoplasm and not translocate into the nucleus. Since Yorkie isn't translocating into the nucleus, there's not going to be the expression of those cyclin or microRNA genes. And that's going to lead to suppressed cell proliferation and the promotion of apoptosis. So that's going to essentially say cells aren't going to divide. If anything, we're going to be losing cells through the process of apoptosis, and it's going to be a result of the overall size of the organ itself or some of the other activating components that we mentioned right up there. As I mentioned before, in addition to observing the Hippo pathway in Drosophila, or fruit flies, we can also observe it in mammals. So it's a relatively similar process, but there are just a few proteins that are kind of swapped out for other homologous proteins within a very similar pathway. So as you can see here, in Drosophila we have the Hippo protein. That's replaced with a homolog MST1 or MST2. And there's also not Yorkie, but there's also a protein complex called YAP and TAZ in the mammalian system, and that's going to facilitate the same function in terms of YAP and TAZ moving in to the nucleus, translocating to bind to DNA, induce cell proliferation, suppress apoptosis, or in the active pathway, when it's phosphorylated, be degraded, and then lead to suppression of cell proliferation and the promotion of apoptosis. The last thing I want to say about the Hippo pathway is that its integration with several other pathways makes it the effector, or what's actually going to change the state of whether a cell is going to divide or undergo apoptosis. And that's going to make it important in connecting everything together and being like the final regulator in terms of whether a cell is going to divide or not. So now that we talked about cell growth, proliferation, intrinsic growth program, and finally a key regulatory pathway, let's tie this all together and answer the question why the child is 50 years old but is still a baby. So in terms of overall growth, I do want to point out the child, Baby Yoda, is 40.5 centimeters tall, and we know a mature member of that species, Jedi Master Yoda, is 66 centimeters tall. So we know that there still needs to be, even though he's going to grow to be relatively small, there's going to need to be some kind of growth to mature him into a full organism. So why is this taking so long? Why is the aging moving at the rate that it does? My theory about that, and it has to do with the timing of cell division, the core of that is Midi-Chlorians. You might be asking yourself, if you haven't watched The Phantom Menace recently, what are midichlorians? These are microscopic life forms that live within all cells. They're able to communicate with the Force and its host, which means that we've got some kind of symbiotic relationship present there. Likely mutualism, because the midichlorians have a host organism, and the person that the midichlorians are living in allow it to connect with the Force. Midichlorians are indicative of being a strong potential Force user, as we know with Anakin Skywalker, as seen in Episode 1. You could see Qui-Gon actually testing his blood for midichlorian levels, like in the video that I showed just before we started talking about midichlorians. And we also know that they're inheritable. When Luke Skywalker says the Force is strong in my family, it's likely that he's referring to the high midichlorian count that's going to pass on to the individual's offsprings. With respect to midichlorians and the Mandalorian itself, we know that this guy right here, the client, who is the person that sends the Mandalorian on the bounty to get Baby Yoda, when discussing with a scientist in Star Wars that's trying to extract a material from Baby Yoda or the child, he says, extract the necessary materials and be done with it. What I'm asserting is that the necessary materials in mention here are actually midichlorians. And those members of this species, whatever it is, we don't know exactly what the name is, as I mentioned, have a naturally incredibly high midichlorian count. And the fact that those symbiotic organisms living within the cells need to grow and develop on their own, which that mechanism is a whole other video in its own. But the fact that those organisms need to grow and develop on their own means that the organism itself, the Baby Yoda or the child, can't grow too quickly. So my theory is that the midichlorians are going to be able to, from the get-go, modify the intrinsic growth program to control the overall size of the organism to begin with, so that they're able to best suit their needs within the organism itself. They're also able to influence cyclin production. So if there aren't enough midichlorians dividing and producing within an organism cell, they're not going to be able to push forward in the cell cycle. So there's going to be some sort of direct connection between midichlorian production and cyclin production within the host cell. And finally, the midichlorians are going to be able to influence the Hippo pathway. So if an organ isn't able to get to the proper size without not having enough midichlorians present, the midichlorians will be able to stop the organ from developing fully until each cell is able to have enough midichlorians to whatever that count's supposed to be as specified by the Intrinsic Growth Program. So that's my thought on why, using biology, that the child is going to be 50 years old but still be a baby on his own. These are my sources. These are where I pulled my images from. And I just wanted to say thank you for taking the time to watch this video. I really hope you enjoyed it. Wanted to say thank you to Brother Albert Garr for teaching an awesome development course that kind of laid the groundwork for this lecture. And I wanted to say thank you to Caitlin Ackclose for organizing our Sci-Fi Fridays. If you have any questions, please send them to my email. I'd be more than happy to talk about this even further. And thank you again for watching. I really hope you enjoyed.